we have most of the bacteria that we're going to be concerned with fall into the phylum, the proteobacteria. And this is very creatively subdivided into 5 classes, kingdom, phylum, class, etc. 5 classes that are just given Greek letters, alpha, beta, gamma, delta, epsilon. So we're just going to take a brief look at some examples in each of these classes and just see what's significant about them. Okay, now there's quite a few here. I will say, just be familiar with one of these. In fact, tell you what, mark this one and mark this one. I realize sometimes there's just a lot of somewhat trivial things to feel like you got to memorize. So in this category, the rickettsia and rhizobium in particular. Okay, a good one and a bad one. Okay, so you can see all 3 of these genera have an important role. They're soil bacteria, so that have some role in the nitrogen cycle in making nitrogen available for plants and thus allowing it to get into the food chain. So rhizobium is of extra interest because rhizobium, it forms a symbiotic relationship with certain food crop plants. The legumes, okay, so legumes, you know, lentils, beans, that kind of thing, as well as alfalfa falls into that group as well. So they're actually associated with the roots of these plants and make nitrogen available by fixing nitrogen gas that's in the air that, you know, percolates into the soil. Why is this worth noting? Because it's the basis of crop rotation. So you've heard of crop rotation? So for example, you know, if the cash crop is corn and the land is used to just grow corn repeatedly and nothing else, you know what's going to happen to that soil? It's going to get depleted of nutrients, okay? Because species like this are not associated with corn plants and corn roots. But if you rotate, meaning, you know, during the summer or during one season you grow corn, the next season you grow alfalfa or something else, or some legume, you're going to bring some rhizobium into the soil. And they're going to create nitrogen compounds that will enrich the soil. So it actually, the rotation of a legume that brings in this symbiotic bacterium will replenish the nutrients, specifically the nitrogen compounds in the soil, and keep the soil fertile over the long term. So rhizobium is very important for that. Okay, and rickettsia is another genus that's important, you know, is relevant to a couple of familiar human diseases, typhus and rickets, also Rocky Mountain spotted fever. And if you're wondering, we will look at select diseases in more detail later in our course. For now, we're just concerned with, okay, so what organism is in what group and what diseases it cause? Just a few examples. The betas. Okay, again, we got a couple of good guys and bad guys. In particular, let's say this one. This one's probably of these two. Let's just say if I ask you about either one, I'll ask you about this one. I'm choosing that one because the name itself will give you a hint as to why it's relevant. Okay. So it's involved in the sulfur cycle. What does thio mean? So thiobacillus, it's obviously going to be it's a name, obviously, it's the name of a genus, but it's going to be a rod that has some relevance to sulfur. Okay? Thio means sulfur. As you can see, they're going to be instrumental in converting poisonous H2S to other sulfur compounds, okay? Sulfate in particular. And that's a good thing. In fact, one organism's waste is typically what another one will use for some purpose. So it can help get rid of H2S buildup. H2S, for example, that's produced by some other toxic organisms. Excuse me. Now, let's look at a couple of these two in particular. So these two are two I'll probably ask you about. Neisseria. Okay. The genus Neisseria, there's actually a second one I could have put in here, but Neisseria gonorrhoeae, okay, cause of gonorrhea. We're all familiar with gonorrhea. I think I mentioned it's an example of, how morphology and arrangement can be very informative. Somebody shows up with symptoms of gonorrhea, do a gram stain on some of the exudates, and if you see gram-negative diplococci, you're probably dealing, something like that. Most likely you're dealing with gonorrhea, because that's a characteristic feature of their morphology and arrangement. Another disease that's on the comeback, unfortunately, is whooping cough. Okay? So the genus is Bordetella. Bordetella pertussis is the species name of the bacterium that causes whooping cough. It actually produces a toxin called pertussis toxin. And that makes it very difficult for the cilia to function and get the mucus out, and hence the difficulty with coughing and so forth. Okay, and don't worry about Burkholder there. Let's move on. Gammas. So we're almost done. The gamma proteobacteria. This is going to have the most bacteria that are familiar and relevant to humans in terms of both good and bad. So here's an example of one that's relevant to ecological health. Methylococcus, as you can see, it breaks down or uses methane. Methane matters these days. You know what methane's significant for? What's that?
Greenhouse gas?
Yes, exactly. Yeah, methane produced by microorganisms, both in soil, water, and in cow guts and so forth. It's a super potent greenhouse gas. And as I recall, it's 20 times more heat trapping than carbon dioxide. So anyways, having organisms that can convert that to something else that obviously won't be so, heat trapping would be a good thing. And again, here's 3 examples. These all you should be aware of. Should be familiar genera. So Vibrio, we all know what Vibrio means now, right? It's a descriptive term for curved rods. So here's some Vibrio cholerae, called that because it's the cause of cholera. They produce a toxin that screws up sodium balance and causes, by osmosis, a loss of water, tremendous loss of water, and literally explosive diarrhea, as they call it, and people die from dehydration. So cholera is caused by Vibrio cholerae. Pseudomonas, we're going to actually, there's a couple of species of Pseudomonas we'll actually handle in the lab. Don't be alarmed. But for example, in hospital environments, species of Pseudomonas can frequently cause infections. They're what we call an opportunistic pathogen. So what does that mean? An opportunistic pathogen? Yeah, it takes advantage of opportunities presented by compromised hosts. Right. Individuals who are sick, weakened, injured, have things puncturing their skin, catheters and IVs and stuff like that. So people in a vulnerable state, which is always going to be the case in a healthcare environment, are more prone to contracting infections by organisms that normally don't cause a problem. So an opportunistic pathogen, for example, people who have immune deficiencies, for example, somebody with AIDS. Somebody with AIDS is going to start getting ill from opportunistic pathogens that you normally come in contact with, but in a normal, healthy state with an intact physical barriers, your skin and so forth, your mucous membranes aren't going to cause any problems. Your defenses take care of them and keep them in check. But people in a compromised state, for one reason or another, can develop opportunistic infections. And Pseudomonas is one of those that's commonly found in hospital environments. And Salmonella. So Salmonella, you've no doubt heard of this. So Salmonella, enterica, typhi, there's a number of species in this genus. This question. Typhoid fever, as you can see, okay, and what we commonly call food poisoning, salmonellosis. And one thing I always like to mention, salmonella is very common on reptiles. You ever heard that? So people with pet reptiles, very, very bad idea to pet your iguana and then peel an orange for your child and hand it to him or something like that. So, you know, if you're ever handling reptiles, always wash your hands before you handle any food. I know not many people have pet reptiles, but some people do. Delta proteobacteria, you know, there's a couple of examples here. These aren't really all that significant for our purposes, so I'm not gonna test you on this, just for the record. Okay, be aware there's this group, and here's a couple of interesting examples, but you know, I'm not gonna ask you about these, just for the record. Okay. So let's just move on to the last group, the epsilon proteobacteria. And we got a couple of examples here to be aware of, a couple pathogens. And so most of them have some sort of curvature. They're rods that have some kind of curvature in the cell, whether it's Vibrio or further, or you know, Spirillum or something like that. So here's a very, very familiar one. H. pylori. Helicobacter is the genus. Helicobacter pylori has now been demonstrated to be the cause of most peptic ulcers, something that was traditionally thought to be due to stress or that sort of thing. But most people are actually exposed to this bacterium, and just in some people, they get sort of a foothold in the stomach, and they can cause local infections that produce inflammation and so forth. They're actually interesting in that they can survive the acidic environment of the stomach. Stomach's, you know, about pH 2, right? When you eat, there's all that acid. And they, in their little local environment in there, they actually produce an enzyme called urease. I'm mentioning that now because you're going to test for that in bacteria later in the course in the lab. But urease, so you've heard of urea, and urea, when you hydrolyze urea, all urea is a little, I don't know if you know what a carbonyl group is, an oxygen double bonded to a carbon, and then a couple of amino groups sticking out. You know, it's a way to kind of, you know, your body uses to temporarily store waste nitrogen, as opposed to as ammonia, which you can't store because it's very toxic to you. So, but anyways, any urea in that location, urease will hydrolyze it into ammonia. So in the stomach where these guys are. And what's the characteristic of ammonia, NH3? What chemical property does it have? It's a base. It's a base, which means it's going to soak up H pluses. So in their little immediate environment, they can raise the pH to be not so acidic for them in their immediate surroundings by producing an enzyme to hydrolyze urea. And so it won't be as acidic for them in their little pocket as it is in the rest of the stomach. And that's how they can get a foothold and survive and start to grow, and then the immune system gets a hold of them or a whiff of them and starts causing inflammatory processes. And before you know it, you got an ulcer. Okay? So but anyways, but for the record, we just want to be aware that that's the main cause of stomach ulcers, peptic ulcers. And one last genus to be aware of, Campylobacter. Campylobacter jejuni, for example, can also cause another form of blood poisoning. All right, so I think this is the end. Oh, I'm sorry. We got one more. And just to be aware of a few other phyla that are gram-negative phyla. Okay, so they have gram-negative cell walls. They don't stain gram-positive, obviously. And again, just some examples of some genera and diseases they cause. So spirochetes, I've mentioned these both previously. There are 2 species of spirochetes that cause very significant illnesses in humans. Syphilis, of course. Treponema is the genus. Treponema pallidum is the species that actually causes syphilis. Quite devastating in the past. Nowadays, very easily treated with antibiotics, provided it's caught in time. And the genus Borrelia burgdorferi, causes Lyme disease. Lyme disease, it's a bacterium. How's it transmitted? Ticks. and you get the little bullseye rash and so forth. And that can be a very significant chronic disease as well. The most commonly transmitted sexual disease, or STD, is we just call chlamydia. Chlamydia trachomatis is the full name of the species that causes that. So, and again, it can cause, especially like in newborns, it can cause blindness and so forth. So newborns always get little antibiotic drops to help in case there's any exposure there. They're kind of interesting. They're also intracellular pathogens. They do have cell walls, but their cell walls do not have peptidoglycan. So there's always little exceptions to the rule. So, we already seen one bacterium that doesn't have any cell wall. Here's one that does, but it's not the same material as most of the others. It doesn't have peptidoglycan. All right, and then last, and I guess not least, Bacteroides is a species of Bacteroides are obligate anaerobes that are very, very common species that live in the gut, the large intestine. And, they're good guys. Okay? Their being there helps you in several ways, helping digest material you couldn't digest otherwise. Outcompeting competitors that could be dangerous, like Clostridium difficile, for example. So anyways, that's our little tour of the bacterial domain. Okay? There's a lot more, obviously, but that'll just give you a few to think about. Okay, so any questions so far? Okay, now just a brief introduction to the archaea. And with the time we have left, probably about 10 minutes, we'll get a head start on chapter 12. And just for the record, the exam will cover all the way up through and including all of chapter 12. So I think we're scheduled to finish chapter 12 on Tuesday, and then do chapter 5 on Thursday next week. Chapter 5 will not be on the exam. Okay? And if we're not quite finished with chapter 12 on Tuesday, we'll just finish it up on Thursday, and all that will still be on the exam. Okay, the archaea. What does archaea mean? What does that sound like? Old, ancient, archaic, archaeology, yeah. So these organisms are thought to be the types of organisms that were the first life on the planet. So they seem to have been around a long, 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 long time. And even though superficially they may have a lot of characteristics in common with the other prokaryotes, the bacteria, they've been now determined to be so different from them. They're given their own domain. It's crazy. Okay? So again, that all comes down to studying at the molecular level, DNA sequences, homology among different genes like the RNA and ribosomes and things like that. So in fact, based on that kind of evidence, they diverged from the bacteria evolutionarily like a couple billion years ago or so. So they've been, in fact, we have more in common as eukaryotes with the archaea than bacteria do. So even though they're both just single-celled prokaryotic organisms. So what's interesting about them to know? Okay, Carl Woese in particular, he gets credit for pointing out that, hey, I've been studying these things, and they're really different. They're really different from bacteria. In fact, we should give them their own category. And eventually he won over the scientific community, and that's the way we classify them. So not only in terms of DNA sequence, but just some of the things they do, or some of the things they have, are very, very different from bacteria. So just vaguely, some metabolic processes are very different, but in particular, ribosomal RNA sequences. Where do you find that? Of course, in ribosomes. And ribosomes are made of proteins and ribosomal RNA. And the sequence of the ribosomal RNA, which is encoded by ribosomal RNA genes, those sequences are commonly used to compare among microbes to determine just how similar or distant they are from each other in evolutionary terms. So it turns out their ribosomal RNA sequences have more in common with our ribosomal RNA sequences than with bacteria. So here's a few others to mention. Translation, which we'll talk about in chapter 7. Okay? Most of you should all have learned something about this, even though every semester when we get to chapter 7, How many people have never had this before? I get a lot of hands. That's okay. If you haven't had it, you haven't had it. Right? You don't know what you don't know. But hopefully your biology prerequisite course talked about how proteins are made by translation and good old gene expression, transcription, and all that good stuff. So something interesting about translation, which you should have had before, is that when a gene is expressed, there's this process called transcription, where the DNA sequence that encodes amino acids is copied into an RNA sequence that also encodes the same amino acids. And then a ribosome gets a hold of that RNA, and it carries out the process of translation. So translation, as you learned it hopefully, every polypeptide made by translation begins with the same amino acid. What is it? Yes, it's methionine, right there. So methionine. And that's the case for translation in your own cells, okay? In eukaryotes, as well as archaea. Bacteria, it's a little different. Probably didn't go over this, but bacteria always start translation of making a polypeptide with a chemically modified methionine. Okay? So the amino acid methionine has an extra little group added to it. It's called a formal group, okay? N-formal methionine. It's little formal groups added to the amino group of the amino acid. Okay? Just one example of how archaea actually have more in common with us than with bacteria. Because archaea do it like we do, bacteria do it their own way. Their cell walls, they don't have cell walls using peptidoglycan. They use a variety of other materials, but they don't make cell, they have cell walls, just not made of peptidoglycan, so made of totally different materials. Their lipids are not the same. In fact, their membranes have very unusual kinds of membrane lipids. So they're not the same as the usual phospholipids you find in prokaryotic membranes or even yours. So the membrane lipids are unique and quite different. And this is just something that's, I hesitate to say it's characteristic of most archaeans, because it really isn't. But a lot of them, certainly not most of them, but a lot of them are notorious for living in super duper extreme environments from our viewpoint. And the early Earth, there's evidence that early Earth had a lot of environments that, is at least with regard to life as we know it today, is very harsh, okay? Lots of acid, very high temperatures, that kind of stuff. High salt concentrations. So like Salt Lake are real high salt environments. There are archaeans that can not only survive it, they require it, they thrive. They need super high salt concentrations that would kill most cells. Others require extreme pHs, like pH 1 or pH 0. They've even found some that in mining waste can thrive in negative pHs. You know what a negative pH is? Because just to remind you how pH works, I'll ask you about pH, by the way, on the exam, I guarantee you. So brush up on that. I don't know if we addressed this yet, probably haven't in class, but a pH, okay? The pH scale, so you see it from 0 to 14, right? What does that number represent? Right, right, so it's referring to the concentration of hydrogen ions, H pluses, or protons, whatever you want to call them. And, but numerically, each number on the pH scale represents what value? 10 to, so for example, say a pH of 8. It means the H plus concentration is what? 10 to the negative 8. And the units are moles per liter. Okay? So I always just tell students the simple thing to remember is the lower the number on the pH scale, the higher the H plus concentration, right? Does that sound familiar? Okay, like a pH 1, a lot more H plus than pH 12. Okay? It's because the value isn't really 1. It's just kind of an abbreviation. It's like a numerical abbreviation. A pH of 1 means 10 to the minus 1 moles per liter of H plus, right? pH 12 means 10 to the minus 12. So really a pH 1 represents a much bigger number than a pH 12. Okay? But people just casually refer to the pH going down, being a lower number, but what it represents is actually a bigger number. And you can even get to negative pH values. So the pH 0 means 10 to the 0 moles per liter, right? 1 mole per liter. What if you have 10 moles per liter? What's that? It's actually a pH of -1. And in certain environments, they've gotten pHs that extreme, that acidic. And they found living cells in there, like archaeans, real fascinating extremophiles. Okay, and then there's also temperatures. And we're going to see a very relevant species later in the course that we call hyperthermophiles. Again, they not only can survive, but thrive in really, really hot environments. We're going to do PCR in a couple of weeks. Polymerase chain reaction, very important technique in research as well as diagnostic testing. And it uses a DNA polymerase produced by a hyperthermophile. And we took advantage of that, so we can do this technique and heat the enzyme to near boiling, and it's just fine. It can keep doing what it does. So anyways, I'm going a little bit on and on here, but a number of them are real fascinating extremophiles. Okay? And here's just a couple of examples. So there is some classification that we can be aware of. Okay, so we have 2 major phyla, and this classification is gradually going to be expanded, and there's actually a few more these days. But most of the thermophiles, the ones that require and love very, very hot environments, are in this phylum here, the Crene-Archaeota. The Euryarchaeota include a lot of the other ones. Okay, halophiles. What does a halophile love? Salt. Okay, very salty environments. Methanogens. Okay, produce a lot of methane. So a lot of that methane we're worried about producing. In fact, a lot of it's at the bottom of the ocean. And if the oceans heat up a little too much, a lot of that can start, it's in kind of a solid form, it can start bubbling up to the surface and increase in the atmosphere. But a lot of methane producing organisms are, we call methanogens. They're in this phylum. And also a few thermophiles as well. And again, for the record, we're not aware of any disease causing archaeans. And let's see, a couple of questions. And then I'll just briefly introduce you to chapter 12. Okay, main features that distinguish cyanobacteria from green and purple bacteria. So that's way back from about 9 o'clock. If you remember any of that. So can you think of some things that are different about them? How about, go ahead. Cyanobacteria, like Anabina, and the purple and green bacteria, the sulfur and non-sulfur ones. What's that? Right, so the cyanobacteria are oxygenic, and the purple and green bacteria aren't, so we call them anoxygenic. And that relates to also where they live and thrive. So the cyanobacteria are also aerobic, right? They better be. They make the stuff, right? But the purple and green bacteria, in terms of oxygen, they're obligate anaerobes. Okay? And I guess that's the main thing. I feel like I'm forgetting something. But and then what they use as a source. So photosynthesis requires electrons from hydrogen atoms. Okay, where do your cyanobacteria get their electrons? From hydrogens in what molecule? Water. And then you have your sulfur, purple-green bacteria from H2S, and your non-sulfur get their electrons from H's from various organic molecules. What phylum do you find most of the endospore-producing pathogens? The firmicutes, exactly. In particular, these aren't the only phyla, but genera, but Bacillus and Clostridium. Bacillus and Clostridium. But again, the answer to that question is the firmicutes. All right, and the last one here, what bacterial genus contains the causative agent of gonorrhea? So it's Neisseria. Neisseria gonorrhea. So try to come up with little word associations to help you remember some of these. That'll help. But Neisseria is the genus that contains the cause of gonorrhea. All right. Now, and again, I hope you're paying attention to these. This isn't the only terms you need to know, but it's just a list of terms that are probably new to many of you. So just make it a point at some point in your studying to make sure you keep those terms straight, because they're going to show up in a lot of questions. And I did explain all this, didn't I? Or did I? This is not required, but at the end of each chapter there's a series of questions. Okay, you have short answer, multiple choice, true, false. Okay. So I'm just indicating the ones that relate to material we actually covered. Okay. All right, looks like you're ready to go. So we'll start chapter 12 next Tuesday. No, that's going to be covered later, after the exam. That part of it I thought it was better to do after